Good morning and welcome to NGI and Nanopore Tech Update Seminar. My name is Susanne Hellstedt-Kaje. I'm head of the long read unit within NGI. And we have experience from using Nanopore since 2018, and where we got our first PromethION sequencer. Today within NGI, we have two Nanopore P24 sequencers, one here with us in Uppsala and one at NGI in Stockholm. On today's agenda, we have several interesting presentations. So first, Jakob Etvik from Oxford Nanopore Technologies will introduce us to nanopore sequencing. After that, my colleague Anna Petri at NGI will tell you about nanopore sequencing at NGI and the experience we have from that. Then we will have a coffee break, and coffee will be served outside here, and you will also have a cookie if you're on site, of course. But after the coffee, we will have 3 research presentations. 2 locals. We have Ida Höjer, who is also a colleague at NGI, but she's also half-time at Clinical Genomics across the street. She will present one of her thesis projects. She defended her PhD thesis last autumn. After that, Carl-Johan Rubin, researcher at Uppsala University and next door to us as well. He has a long experience from using nanopore in his research. And finally, Melanie Tanguy from Genomics Inland will zoom in from the UK, and she will present her experiences from sequencing whole genome cancer samples. We will end today's seminar with a Q&A session. It will be possible to ask questions directly here in the room, and also if you use the raise hand function, we will let you in and you can ask the question directly. If you have questions to the speakers during the presentations, feel free to use the chat box, but make sure that you write to Anna if the question is to Anna, for example, because if we have time, we can take one question after each presentation, but if there is no time, we will take the questions at the end of the Q&A session. So then it's important that we know who the question was directed to. So that's it from me. So I will leave the stage to Jakob to introduce nanopore sequencing.
Thank you. That looks right. Good. Thank you, Susanne, and thank you very much, NGI, for inviting me to speak here today. And good morning, everyone. I'm just going to give you a very brief introduction to nanopore sequencing, the very basics of it. And then I'm just going to show you a few snippets of what people have been up to lately, as well as some of the latest technology updates that we have launched. And I have so much to say, so I'll try to keep within time, but I put my time on as well to keep track of that. But first of all, the mission with Oxford Nanopore is really that we want to enable anyone to sequence anything, anywhere. And while we're not quite there yet, we have seen people applying nanopore sequencing in loads of very exotic places, such as the International Space Station, deep sea expeditions on glaciers, etc. Not as pleasant, but recently over the pandemic, we've also seen people implementing nanopore sequencing for whole genome SARS-CoV-2 sequencing for epidemiological variant monitoring across more than 85 countries. And this is, of course, in standard labs like you would have here in Sweden, but it's also in more rural districts across the third world countries, etc. And we love to celebrate the work of the Nanopore users in the community. And in particular, last year, we saw a major uptick in terms of the published research studies. So these data are from November last year, but I think we actually capped just around 1,000 new publications over 2021 alone. And yeah, so much exciting stuff is ongoing. But in terms of nanopore sequencing, I assume many of you have seen this image by now that I'm going to show, but the very basics of it is that a DNA or RNA strand is passing through a nanopore, and then an electrical signal is generated, which is then interpreted into base sequences. So a cross-section of a nanopore flow cell would look something like this, if you were to draw it in a cartoon at least. So it consists of a few different modalities. So you have the sample DNA or RNA, and then as part of the library preparation, this purple motor enzyme is connected to the DNA strand, and then the nanopore sits within these synthetic membranes on the flow cell. And once the nucleic acid passes through, there is a current signal generated, which our base calling algorithms then can translate into base sequences. And there are a couple of other important parameters in the overall system, of course. The data is instantly generated, and you can do real-time base calling as well. So it's also a general feature of the technology that it works in real time. So why nanopore sequencing? There are a number of different benefits and omics capabilities that we like to mention. So first of all, it's direct sequencing of DNA and RNA as the only platform that we are aware of. So it's a single molecule from your original sample that you can analyze directly. So of course you can do PCR or other types of amplification if you like to, but in a lot of applications it makes more sense to just read the native strands. So there's no sort of lasers or other things involved in the detection. It's just this signal with the current changed through the nanopore. It also means that if you read the native DNA and RNA, you get the base modification signal as part of the raw data. So if you're interested in studying the epigenetic part of your biological sample, then that's sort of an inherited part of the raw data. And then also, the system will read any sample you present to it, so it doesn't have any limitations on the read length generated. So it's down to the sample you provide and the biological question, which helps you guide what type of read length you would be looking at. So as of last week, we just extended the range ever so slightly. So previously we used to have a sort of a synthetic, or at least just a chosen cutoff of around 200 bases at the lower end of the spectrum. But last week we released the short fragment mode, so now you can sequence down to 20 bases. For the time being, so this is not a strict limit or anything, but the current record is more than 5 million bases. So in the current scheme of things, you can sequence anything from 20 to 5 million bases per single molecule read. And as mentioned before, it's also in real time. So if you do real-time base calling, you can start analyzing as you go. And there's also the option to start and stop flow cells as you like. So if you collected enough data after 6 hours or 24 hours, you can just stop the flow cell and reuse it, wash it and reuse it for something else later on. It also gives you the option of adaptive sampling, which is an on-sequencer target enrichment method that I'll just speak a little more to, as I think Anna is going to speak a little bit about it as well with the local experiences. So it's a pretty neat methodology, because we can, the hundreds to thousands of nanopore channels within each flow cell, we can control individually via the software. So you don't have to do that manually, of course, but it's now built into Minnow, so you can do that straight off of the system. So whenever a strand starts sequencing, then using the adaptive sampling just means that you bioinformatically defined regions of interest that you want to analyze later on. So once you start sequencing, the DNA is real-time base called and real-time aligned to your reference of choice. And if the sequence is then within that specified region, it'll just continue sequencing through. And once it's complete, the nanopore will be available for the next strand of DNA. Contrary, if you start sequencing and the real-time base calling and real-time alignment shows that this is not a region of interest, then we'll reverse the current, or the system will reverse the current and spit out the strand after approximately 1 second, having read around 400 bases. So with the exact same library preparation method, you can generate bioinformatically target enrichment in a quite elegant way. And again, if you're using native DNA, you also get the base modifications directly relative to doing PCR enrichment or similar. So just to mention the scalability of the technology, the exact same sample prep, same sequencing chemistry, and same data analysis pipelines are applicable across the board. So these are the commercial systems we have available today, down from the fungal flow cell type, which would typically give you 1 to 2 gigabases of data per full run, and then all the way up to the PromethIONs, of which the PromethION 24s are what Susanne mentioned is here at NGI already. And these all come in these starter packs, so it's extremely accessible to everyone, because it's sort of built in with the loan device and also upgrades down the line. In terms of tech news, I chose a few highlights, which I thought was mostly relevant for you guys here. So these were all presented in December at our Nanopore community meeting. So these are the headline numbers. So these are the benchmarking data that we put out there. And as you can see, looking at the full genome, including repeat elements like centromeres and telomeres, we now have a very high raw read accuracy. So again, the single molecule, or on the single molecule level, you can now achieve Q20. And we introduced duplex reads recently, which is in early access still, but with that you can get even higher. And in terms of SNP detection and structural variants, etc., it's also looking very nice now, and we're getting extremely close to short read performance metrics. And we also provide open data sets. So if anyone is interested in understanding how we achieve these benchmarks, it's freely available to download, including guidelines and on the data analysis, etc., and how we've done it. So feel free to have a look if you want. And just to highlight a few of the different parameters we do to continuously improve the technology, includes our base callers and algorithms in particular. So the depth graph here just shows the development over approximately 2 years in the past, where the raw read accuracy improved from around 92% to 98% just by improving the machine learning of our base calling algorithms. So that's one parameter we can modify. So we also have the actual pore types, we have the chemistry, we have the membranes, etc. So another parameter we're currently working heavily on is the chemistry itself. So where the pore type 9 and the KIT 10 are the current production models, we introduced early access on pore type 10 and KIT 12. And with that, we see these Q20 single reads for now. And another thing which was presented at the Nanopore community meeting is the now improved base calling for plants specifically. So we learned that plant-specific modifications could actually end up showing sequencing errors rather than the modification. So with a specific plant training set, we could minimize that that error mode quite dramatically. So it's exemplified here where for a specific plant data set, we actually saw a very dramatic accuracy increase after the plant specific training. And we have a number of users who have already implemented this for and actually co-developed some of it with us for generating much better plant genome assemblies as well. And I'm almost out of time, so I'll speed up a little bit. But we also have news on base modifications. So whether you use KIT 10 or KIT 12, it shows extremely good correlation with whole genome bisulfite sequencing. And again, it's part of the data you generate already with the base sequences. So we really see a bright future for anyone wanting to do modification analysis with Nanopore as well. Real quick, we have a new PCR cDNA kit, which will provide extra features within expression analysis and also isoform differentiation and splice mapping. So it's just coming out in early access now. But again, I'm happy to speak to you individually about this afterwards, because again, there's a bit of a time limit right now. But anyhow, just a few snippets of what people have been using Nanopore for the first 3 months, well, and probably before, but at least have presented on for the past 3 months in 2022. So again, just a few examples. So one being Danny Miller and colleagues generating a really high assembly quality from just 190 nanogram, sorry, of DNA from fruit flies. Also, Ira Devison from the Garvin Institute and others setting adaptive sampling up for neurodegenerative diseases, where they could assess, I think, across 50 different repeat disorders in one go. And also something quite different where an Australian group developed this tail hair technology so they can assess the age of an individual cow for improved cattle herd performance in their agriculture. One very exciting recent publication is from John Gosinski and a lot of other people out of Stanford in a collaboration with Google, NVIDIA, and Nanopore, where they broke the record of the fastest clinical human genome sequencing. So specifically for critically ill children, and they achieved this in less than 8 hours from sample to clinical report. So it's really exciting to see how far they've come with that already, and they're continuously working on with it. So I'm out of time. So with that, everything I just said is news from December, and actually our CTO Clive Brown will present on a seminar later today the latest development, presumably talking about the Q20 chemistry and other new exciting stuff coming around. So so if you want to hear more, I can highly recommend you to tune in or watch it afterwards. It's going to be available online as well. So with that, thank you very much, and I'm happy to elaborate on anything during Q&A or afterwards.
Thank you very much, Jakob. Great to have you here. I think we were a little bit short of time. So, but is there any quick question? Is there a quick question in the room maybe that we can take? while Anna is getting rigged up for the talk. Yes, we have one.
Yeah, on the target, I don't know if it was kind of target sequencing, but the one that you can select which regions you want to sequence, and if not, the pore will just expel out the read. Does it affect all the pores, or is it only for the pore that doesn't have the correct DNA when you change the electricity?
So it's can people hear me when I speak here, or?
Yes, but it's better with a microphone, so please take them.
Right. So the question was on the adaptive sampling part and whether the individual nanopore is controlled or whether it's across the flow cell, right?
Yeah.
Yeah. So it's actually quite elegant. So you can divide the flow cell into two different parts. So half of the flow cell you just run regular without adaptive sampling. The other part you can run adaptive sampling on. So you can compare directly in one flow cell how your sample performs with adaptive sampling turned on versus not. But it is the individual nanopore channel which is controlled by the software. So you reverse the current specifically on that strand passing through a single nanopore and looking whether it's within the region of interest specified.
Thank you.
Okay, nu är det jag som hörs. I'm the one to be speaking now. Hello, and nice to see at least some people here on site. It's a very exciting news since the last 2 years to actually be speaking in real life. My name is Anna, and I'm working here at NGI with nanopore sequencing. And I will be talking, like Susanne said earlier, a little bit about our more sort of down to ground, like wet lab experiences with nanopore. And this one is falling off actually. My ear is too small. Okay. Yeah. Otherwise I'll just have to hold it. So just jag går inte det här. Men jag måste ha på fel. Nu. Okay. This is not my computer, so some technical issues. So just like Susanne said, in NGI, we have 2 nodes in Stockholm and here in Uppsala. And we have 1 PromethION sequencer each. And then we also have a bunch of MinIONs. The MinIONs are mainly used for, say, QC and like smaller projects. There's also Flongle inserts into these Flongle adapters. So they can be used for like pilot studies and stuff. But the PromethION sequences are more like production machines. The most commonly used method here at NGI is the ligation library prep, in which basically you just very shortly you ligate the sequencing adapters, like you can maybe see on this very bleak slide. On the sequencing adapters, there are motor proteins, and these are feeding the DNA through the pores, like you've already seen on Jacob's slide previously. And these are mainly then with us used for genomic sequencing. We feel that this works well on most samples, probably because there's a lot of things you can do while working with the DNA. For example, there are several purification steps during library prep and also before library prep. And input DNA can be shared to increase throughput. This is because DNA in solution, it coils up, it forms secondary structures, more or less depending on the genome, but it always does. So when they coil up and form these secondary structures, they will get sort of stuck in the nanopores and the pores will be blocked. So if you share the DNA, this will happen less frequently and you will have more data. And there's also more DNA fragments if they're shorter, for reasons that you probably already know. So there's a higher chance of the fragments actually finding the pores. You can think of the nanopore flow cell as, I don't know, a huge landscape with a few distributed pores here and there, and then like the DNA is searching for the pores. If there's more, it doesn't find the pores so easily. Although I did say that we can share the DNA, this is not always what you want, of course, because a lot of people are after the really long reads that Nanopore can master. And this is obviously absolutely doable. What you can do then is that you can load your long DNA and you can let it, you can load your long DNA and you can let it just clog the pores for a while while sequencing. Then you can wash the flow cell with nuclease and in that way degrade the coiled up DNA. And then you can reload your library and have more of it. So it's kind of a stable method in this way. Typically, when we work with this, we like to remove the really shortest DNA fragments because most of the users are after the long fragments. And if you keep the short fragments, they will dominate the sequencing run, like in most other sequencing techniques. So how much then DNA to provide to us and what kind of DNA should you provide to us? Well, this is a project discussion topic. It's not something that I can just answer very easily here and now today. But just as a very rough guideline, say something like 5 micrograms of DNA. If you need really long libraries, like say 100 KB libraries, there might be more needed. If you can't master that much, it's not a problem either. We can solve it somehow. It might not be so long, but there will be something that you might be happy with. I have written here 5 micrograms of not too fragmented DNA, and this is because I would like to make a point of this. If the DNA is really fragmented, we will have a hard time to take away the short fragments and still have enough left for sequencing. I want to show an example of this. This is from one project that we were working with recently here. So first, the sample, this is the femtopulse profile of 2 samples, but extracted from the same individual. So to the left, you can see that the sample is fairly degraded. This was the one that was first submitted to us. And it wasn't also that much DNA present in this sample. It was hard for us to have enough long fragments left before even starting library prep. And when we did start library prep, it basically did not work to sequence. So this was just a big no. We didn't even load this one on the PromethION. We just instead asked for a new sample. They redid the extraction, took some more care, had some more DNA. And as you can see on the right side, it's far less fragmented. So there was no problem with this one. It gave beautiful DNA with good quality and lots of data. So just there's only so much we can do here with our library prep. Input DNA is really the key. I think my colleague Olga has been saying this a million times already, but it's really, really true. So apart from not too fragmented, it's also good to have pure DNA. If you imagine this classical picture that Nanopore usually likes to present of like the 3D version of their pore with a single-stranded or a double-stranded DNA being untangled or being turned into a single strand and fed through the pore. This is like a happily ongoing process until you add some contamination. This is a very over-dramatized picture, but you just get the point here. So if this contamination is big enough and bad enough, it gets stuck in the pore and the pores are blocked and will not be available for sequencing anymore. So try to make sure to have pure DNA. We get lots of different samples here at NGI, obviously, and we find that perhaps not so surprisingly, human DNA is often the most easy to work with. People often ask us how long, they come to us to ask how long should library be? And we have in some studies found that, obviously depending on your question, but for most like structural variant studies and maybe like spanning difficult regions, a 20 to 30 KB library may be a good trade-off between the length that you're needing and the throughput that you're needing. Obviously you can have longer than this as well. But this is like the 20, 30 KB library, then you will have lots of data from just one flow cell. Possibly also not so very surprising is that plant DNA is often more challenging to work with. There is a lot more contaminations in plant DNA, such as secondary metabolites and stuff. But there's no reason to give up. You can always submit plant DNA to us. Again, if there is, like you see on the top picture, a contamination stuck in the pore and stuck on the DNA, you can wash with nuclease, and in many times the pore will actually be free again for sequencing. It doesn't degrade the contamination, but if the contamination is stuck on the DNA, it sort of helps anyway. And I would like to point out, now Jacob has already told you a lot about the new Q20 or the new 20 kits, which are very promising. But even without these, we have during the last year seen great improvements from Nanopore in terms of both kits and flow cell stability and also base calling quality. So we get better data and more data, and it's easier to get for us now than it was only just 1 year ago or even half a year ago. So this is a happy time to work with Nanopore. So I've been talking now a lot about genomic ligation sequencing, but you can argue that there is so much more you can do with Nanopore, and this is true. We don't have plenty of time today, but I will just briefly mention some of the experiences that we have done here at NGI. One very interesting application from Nanopore is the ultra-long sequencing. Like Jacob already said, there's no limitation, at least there's no theoretical limitation to the read length that you can have. So they have developed a kit to acquire megabase long reads. In this kit, I know this picture is not fantastic, it's a bit bleak as well. But in this kit, you just add transposase to your DNA, and then where this transposase cleaves randomly, you add also adapters, and then you just load that on the flow cell. There's a short cleanup, but it's not very big. The DNA will pass through the pores for a while, and then it will clog, like long DNA does, and then you will wash it with nuclease and reload. So if you have good DNA, you will have really long libraries, read libraries from this one. But there is not much happening during the library prep. So if DNA is key in other library preps, this is even more key here. It really needs to be pure, and if you don't have megabase long DNA, obviously, you will not have megabase long reads either. This may be a bit discouraging for some, but I have some comfort for you. We have lots of experience with DNA extraction here at NGI, and we also have an extraction service I can just mention briefly. So for projects that are sequencing here with us with long read technologies, we can do some DNA extraction as well. So just come discuss this with us if you need. Jacob has already mentioned DNA methylation, and this is becoming more and more common with Nanopore now. Maybe it's, I might not say gold standard, but it's almost becoming gold standard in DNA methylation with Nanopore. It's the base modification are always part of the raw data, like you've already heard. So it's just a question of like extracting this information from the raw data. And this is kind of a focus now on Nanopore side. You don't need any special library prep for this. Whatever library you're loading, you can have your raw data information from. So we tend to just use our standard ligation prep. We share quite much if it's just a pure modification project, so that you can have lots of DNA and high quality and good coverage. The problem is instead then with the actual base calling, you need lots of graphic cards, which are kind of high tech for this, lots of GPU power. This is also getting better, both on our side and on Nanopore side. You can obviously do cDNA sequencing with Nanopore. There has been some experience with that in Stockholm. The cDNA protocols, they convert RNA to cDNA using strand switching, which means that they enrich for full-length RNA molecules, meaning capped molecules. And these are then obviously also sequenced, like there's no length limitation, and this might add a lot of value to your isoform analysis. It's also very well suited for spatial transcriptomics, and our colleagues in Stockholm have been using this in combination with the 10x VSU method. You might not know what that is, but it's just a method for spatial transcriptomics, basically. It's also well suited for single cell analysis. And if you're interested, there are also PCR-free options. I would like to mention this direct RNA sequencing. It's kind of a niche application still, but it's unique to Nanopore that you can sequence native RNA. And then also, obviously, you have the base modification. So if you want to study base modification in RNA, this might be a good way to go. It works in the way that your RNA molecules are stabilized with a synthetic cDNA complementary strand so that you have a hybrid molecule. And the sequencing adapters with monoproteins are then only ligated to the RNA part of this hybrid molecule. And then these are the only ones being fed through the pores. Like I said already, it's a niche application. It's maybe not for everyone. It's very demanding on the RNA input. There has to be a lot of it, and it has to be really pure, actually. Also, once you have the data, the base calling models are not prioritized currently from Nanopore side, so you will have to use third-party tools. And you might also have to train your own base calling models to fit your data, fit your sample. But it's fully doable, so if you're interested, you can register such a project to us. And finally, I want to talk about targeted sequencing. It's something that I find really interesting personally. It's like a dream, I think, for many researchers to sequence only a part of your genome without using PCR that messes everything up. And there has been lots of methods developed over the years, like hybrid captures and things that have never worked like according to your dreams. Nanopore has developed two methods which are really promising for this. One of them is using this fantastic gene scissors that we've heard about. They got the Nobel Prize for a while ago. So in this, again, a very bleak picture, you can see that you use guide RNAs to, you design your own guide RNAs to target your region of interest, ROE. And then you use this CRISPR-Cas9 complex to cut out those regions, and then adapters are only likely to what is being cut out. There's a separate kit for this one, so it requires a lot of extra work, both when designing the guide RNAs and when doing it in the lab. But it's pretty well working, and it gives pretty high target coverage. The other method you have already heard about, and that is called adaptive sampling. It's very promising. This is a regular ligation prep, or actually a regular prep, whatever you want to do. You don't have to do just a ligation, you can do anything else. We have done it once here, or Ida, my colleague Ida Heijer has done it once. Saying that we have experience with this is, well, we have some experience from one experiment. What happens then, this is a picture of Ida loading her sample on our new Milan computer. So what you do is that you load your regular genomic library, preferably a ligation library, which is good. And into the computer then, which runs your sequencing, you load a FASTA file with your target reference. And as you've already heard from Jacob, while sequencing is ongoing, if the read does not match your target, it will be ejected from the pore by reversing the voltage or reversing the current in that pore, so that the pore is available then for new for new reads. And this is quite interesting. And I can also say that since this picture was taken, we have a longer wire for the MinION so that it reaches down to the bench now, and Ida can in the future load her MinION on the bench sitting on a chair like normal people do, and not having to stand on an elephant foot with the MinION on an upside down waste box. This is like a temporary solution. So I think these two targeted approaches are kind of interesting. So I've been allowed to present very, very shortly just some of her results. Ida has been working during her PhD with Cas9 enrichment quite a lot. One example from that is when she was studying the FLG gene, which is if you have mutations in this gene, it is associated with a skin disease. And it's a very complex region, which is, as I understand it, then responsible for these mutations. So it has not been possible to sequence before. There's a lot of repeats, 10 to 12 kb repeats. And as you can see on these pictures, there is one patient with 2 alleles, and one normal allele, 1, and the other allele 2. 2 repeats have been duplicated, repeat number 10 and repeat number 8. And to my understanding, Ida wasn't the first one to be able to sequence this as a whole. So this is just a fragment of a coverage plot of the entire target. So what you can see here is actually 1, 2, 3 cleavage sites on each side. 1, 2, 3, so meaning 3 guide RNAs. And a lot of good coverage within the target. You can also see a very few off-target reads. So I would say this is kind of a successful experiment. But obviously the off-target reads are also very important, and this is what Ida will be talking about during her talk after coffee. So results from her adaptive sampling that you saw loading in the previous picture. It's just basically it works. And there's not much results from it yet because it was done just a couple of weeks ago. But it does work. So she was enriching for the BCR gene, which is associated with cancer in humans. Well, it works. That's good. We will do more. We will be doing more work with this. So finally, I would like to just say something about what we are doing in the coming months, like 1 or 2 months. If you are interested in like approaching us again in 2 months, we will have more experience with targeted sequencing, obviously. We will have done more cDNA sequencing. These are like projects that we have in the pipeline currently. And we will also bite into low input protocols where there's a choice of amplification methods. And perhaps most excitingly for us, we want to have more GPU power to be able to base call better. We have some in the lab already, but we are looking for more. And we think that we're kind of there, kind of soon. For this, we will be doing targeted sequencing with adaptive sampling, and we will be able to sequence more samples in parallel and also do base modification calling during sequencing. Last, but perhaps not least, I would just like to push for SciLife Labs national bioinformatics node, NBIS. They can also provide support with Nanopore data. Some examples that they want to highlight is that they have pipelines for de novo assembly and annotation, specific for Nanopore data, for structural variants, and for isoform analysis from both RNA and cDNA. So if you want to contact them, you can go to their website, nbis.se, or via the portal when you order a project from us. And this is all from me, and you are welcome to ask questions during the Q&A session or coffee.
Thank you very much, Anna, for a nice presentation. Are you really, do you want coffee or do we have time for one question? I think we have one question here in the chat for you, Anna. Yeah, maybe we take that one now. So, actually we have two, but we take one of them. Anna, so the question is, do you modify the standard ligation library prep when working with large or larger, well, 10 kb DNA, and not do impure cleanups, or use alternative cleanup methods between steps to avoid DNA loss?
Yeah, that's a very valid question, actually. The answer is basically no, we don't. There's a lot of alternative purification methods that you can use, but as a service provider, we need to sort of follow protocols from Nanopore. And as long as they haven't released a protocol for other purification methods, we don't really have a case against Nanopore if it fails. If things fail, we can approach Nanopore and say, we did just as you said, but it didn't work. Can you do something about this? Obviously, if there's like a research project, and I mean, in rare cases, we might be involved in doing modifications, but not on a standard basis. No, sorry to say that.
Okay, thank you very much. So we leave the second question for the Q&A session at the end of this seminar. So now we go for coffee, and we start again at 10:40. See you then. Okay, we start again. So now it's time for the research presentations, and first out is Ida Höjer. So please go ahead, Ida.
Hello, hopefully you can hear me now. So as Susanne said, my name is Ida, and I recently defended my PhD thesis. It was just a few months ago. And currently I share my time between NGI and clinical genomics here in Uppsala. And today I will present to you a little bit of what I did during my PhD regarding studying the effects of CRISPR-Cas9 genome editing while using long-read sequencing. So first of all, I just want to give you a little bit of a backstory on how we actually started with studying CRISPR-Cas9 off targets. So some years ago, early in my PhD, we published this paper where we used the Cas9 enrichment, but with smart sequencing to study repeat expansion in the HTT gene, which is associated with Huntington's disease. And while we did this, we enriched for the HTT gene, but also some other genes in our sequencing assay. And these are the colorful peaks right here. So this is just a sample from our project a few years ago. And this is a coverage plot for one of the samples where you can see the enrichment throughout the genome. And as you see here, we had enrichment for the genes we actually wanted to enrich for, but we're a little bit curious because there seems to be other places in the genome that were enriched for as well. And when we looked more into this, we actually saw that some of these peaks corresponded to what seemed to be off targets caused by the guide RNA sitting down in other places in the genome and enriching for these as well. And this is basically caused by that the guide RNA sequence is very similar to other locations in the genome, and thus you also enrich for these regions. So we got curious, like how can we further develop this method to use it to actually study Cas9 off targets? And the first question then that we asked ourselves was like, why do we need to study Cas9 off-target effects at all? I mean, I think the most obvious answer to this question is that if you do genome editing and you also have off-targets, it has the potential to disrupt genes or other regulatory elements in the genome. And this could be, of course, of particular concern if you're using it for clinical applications. And then the second question was like, how could a long read sequencing assay be beneficial in this case? Because there were other methods out there that were using short read sequencing to do this. But we argued that since we're using long read sequencing, we will also use the benefits of long read sequencing. We will have no PCR and thus no PCR bias, and we can also study repetitive regions or regions with a low or high GC content and be able to actually detect the Cas9 off targets in these regions as well. So we did this, and in 2020 we published an article describing these new methods that we developed, and the first one was based on this smart sequencing method that we used in the HTT paper. And we call it Smart OTS, and OTS is for off-target sequencing. But also, we developed another method for nanopore sequencing that we call Nano OTS, that we found to be really fast and really robust way to approach this problem. I'm not going to go into detail how this method works. The important part is here that you use the guide RNA in this method that you want to use for your genome editing studies. And you will cleave your DNA in this assay. And what you will end up with is a read pattern when you map your reads later that looks like this. So where the Cas9 has cleaved your DNA, you will have like a really sharp line in your mapping profile where the reads pretty much start or end at the exactly same position. And this is something that we use in our data analysis and how we can detect the on targets, but also all the off targets. And we also wanted to demonstrate how we thought that long read sequencing would be beneficial when you're doing this kind of assay. So we actually designed some guide RNAs to target something called dark regions in the genome. And dark regions are typically referred to as regions where short read sequencing has a hard time to obtain any coverage at all. So on the bottom here, this is an example for a guide RNA that we designed for the OPN1LW gene. And on the bottom, you see whole genome sequencing Illumina reads. And these red boxes are these so-called dark regions. And we see that we don't have any reads here from the short read sequencing data. This was also described in a publication by Ebert et al. In 2019. And on the top, we have the results from our Nano OTS assay. And you can see our Cas9 cleavage site where we target our guide RNA to sit down. And we can see that we had no problem in detecting this Cas9 cleavage site, and also that we have reads that span this entire region. And in addition, this was the actually on target that we designed for, where we were also able to detect off targets for some of these guide RNAs that were also in other dark regions in the genome. So this shows that the long read sequencing assay for detecting Cas9 off targets could be very beneficial if you're using one of our methods. So this was, now we had these methods and we did these in vitro assays, but we also were curious what actually happens in a vivo system when you do genome editing. So we wanted to follow up our previous study to do a little bit of research in an in vivo system, and especially to investigate the prevalence of structural variants when you do genome editing and use long read sequencing to see what's going on in those genomes. So we did some genome editing in zebrafish, and we studied on and off targets. And we also did this over generations to see if something is passed on to the next generation. And as I already mentioned, we solely used long-read sequencing in this project. We used NanoOTS to screen the guide RNAs that we were using in the genome editing experiments to detect the off targets. And then we used large amplicon resequencing of the on-target sites, but also for all the off-target sites that we found with our NanoOTS methods. And for some of the samples that we had, we also did some Nanopore whole genome sequencing for some further investigation, but I will come back to that in a little bit. And this study we actually already published a couple of months ago, so in Nature Communications. So the rest of my talk will be showing some of the results that is included in this publication. So first of all, I just wanted to give a little overview of our experimental setup for our genome editing experiments in our zebrafish. So what we first did was to do genome editing of fertilized zebrafish eggs at the one-cell stage. So we microinjected the ribonucleate proteins to do the CRISPR-Cas9 to produce a genetically modified F0 generation. And then we had some random incrossing of F0 individuals to produce an F1 generations. And throughout this experiment, we collected samples for analysis. So first of all, we collected some pools of uninjected zebrafish that we used as controls. And we also collected some pools of F0 larvae. This is partly because we wanted to get a more comprehensive view of a larger sample set, but also to see what happens in an early stage in the development. And we also collected individual F0 adults to be able to study single genomes, but also to see what's happening in a later stage in the development. And then for the F1 generation, we collected pretty much the same samples with the pools of F1 larvae, but also individual fish. So we selected, we chose to work with 4 different guide RNAs when we did our genome editing experiments. So we did 4 parallel genome editing experiments, and we screened all of these 4 guide RNAs with our NanoOTS method to detect the off targets. These genes have previously been used in other functional studies, and they're genes that are associated with they're orthogonal to human genes that are associated with cardiometabolic diseases. The genes that we chose to target are LDLRA, MBL2, SH2B3, and the YWHAKA gene, which I will from now on call Oaxaca because of the simple way to pronounce it. So we did our genome editing experiment, and then the first thing we did was to do large amplicon sequencing of all the on-target sites, and this is in the F0 larvae, but also we did amplicons of all the detected off-target sites from the NanoOTS assay. And on the left here, you see this is plots that show the editing efficiency of these guide RNAs. On the left, you have the editing efficiencies for the on-targets, and for all the 4 different guide RNAs, the editing efficiency were over 90%. On the right, you see the editing efficiencies for all the off-target sites. And what we could conclude from this was that we actually only saw real off-target activity at 3 of these off-target sites. So for the further analyses, when we showing data for the off-targets, it's only from these 3 off-targets. We also looked a bit about the size distribution of the Cas9 editing events, or the Cas9 mutations that were induced. Most of the variants that we saw were small insertions and deletions, but we also saw that a proportion, in this case, 7% of the on-targets were according to larger structural variants. And when I say larger, in our case, we mean variants that are over 50 base pair in length. And the plot here is showing the distribution and the size of every single event. The minus axis are deletions and the positive ones are insertions. So we see that we have deletions that we found that were over 5 kb in size, and also insertions that were well over 1 kb. And for the off targets, we see something very similar. Again, most of the events were small insertions and deletions, but also a proportion of them were larger, with some variants that were over several kb. And I just wanted to show you one example of how it looked in one F0 individual. And this is an off target for the Oaxaca gene. And this is off target is in the Oaxaca B gene. And in this individual, we saw a deletion that was almost 1 kb in size, and it spanned actually an entire exon in this gene that was an off target. We also looked into the F1 generation, and we saw that also these larger structural variants were passed on to the next generation. And I'll show you a couple of examples. This first one is an on-target for the SH2B3 gene. And we saw a deletion here that was over 1 kb in size, spanning almost an entire exon. And also this event seemed to be homozygous in this fish. Another example is an insertion of almost 300 base pairs at the on-target in the Oaxaca gene. And this also seemed to be homozygous. And this was also something peculiar to us, because when we looked at these F1 individual fish, actually we saw some kind of editing event for all the alleles that we studied. But for a portion of the fish, they also seemed to be homozygous. And we were asking ourselves, are these really true homozygous events that we are seeing, or are we missing something? Is it either because maybe a larger rearrangement or a larger deletion that our amplicons might not be picking up, or could be due to some other biases in our analysis? So we went ahead and did some whole genome sequencing with Nanopore for some of these fish. And I will show you 2 examples of 2 of the fish that we found. So this is a picture showing the first allele of these 2 individuals. And this is data from the amplicon sequencing. So the first one had a 21 base pair deletion at the on-target site, and the other one a 3 base pair deletion. And as you can see, almost all of the reads were supporting these allele. And then when we did the whole genome sequencing, we actually found a second allele as well. And the reads were distributed almost equally between these 2 alleles. So it seems to be a really heterozygous variance here. So in the first fish, we had an allele with a 6 base pair deletion, and the other one a 5 base pair deletion. So then we went back to the amplicon data, and we looked at those few reads that were not supporting the allele 1, and we actually saw that, okay, we can see this other allele here as well. But we asked ourselves, like, well, why didn't our amplicon sequencing data pick up these alleles? So we did some further digging, and what we saw was that we actually had an insertion, pretty big insertion of a repetitive AT region. And when we looked in our amplicon data, we saw that a lot of the reads here were terminated at these repeats. So the PCR didn't seem to be able to actually go through this repeat and amplify this allele. And this is why we had this drop of the second allele when we did our amplicon sequencing. So this is also an important finding, that even though we used long read sequencing and amplicon sequencing, this in some cases might not be enough to get the fuller picture of what's happening in the genome. And just to conclude and summarize this last project, we were able to find large structural variants, both at on and off target sites in our zebrafish, and 7% of these events seem to be were larger, were structural variants that were over 50 base pairs at the on-target site. We also found large structural variants were able to segregate into the F1 generations. And I think the take-home message here is it's very important to choose the proper and most relevant method when you're doing validation of your genome or your CRISPR-Cas9 genome experiments, because a lot of what I showed you here probably would have been missed with conventional Sanger sequencing or short-read sequencing, but we were able to find using alternative long-read methods. And this also reason that we actually want to propose when you're doing genome editing validation is we think that you should do an in vitro based off-target detection method like our NanoTS, which is also very easy to use and very fast. But also when you do your validation of your genome edited cells or organism that you're working with, that you use long-read sequencing. And in many cases, amplicons might be enough, but if you find something a little bit peculiar, maybe you have to resort to whole genome sequencing to actually see what's going on. And with this, I just want to thank everybody that's involved in these projects, especially Adam, who did a lot of work with these projects, and also Marcel Denhöd and his group, Anastasia, who we collaborated very closely with the zebrafish genome editing experiments. And lastly, it's nice to actually be able to speak in front of a real audience again. Then I think a lot of people are on Zoom as well. But yeah, thank you.
Thank you very much, Ida, for a very nice presentation. I have a question for you here from the audience. Let me see. So the question is it possible to add topoisomerase enzyme to detangle? Is the question.
I don't know where that isn't that for Anna?
Yeah, maybe Anna.
I don't know if that's related to my presentation.
I will send the question to anyone in the room. Jakob, do you have can you comment? You can go, you have the microphone, so you can.
All right. I'll just speak to the it's a very good question. We have been doing some work with a few, I think, Nanopore users we're working on a topoisomerase based chemistry, but it's still in the development phase, so it's nothing that's currently available, but it's definitely something which could potentially be a product in the future. But right now, I don't think there's an easy way to apply it to topoisomerase to untangle DNA and increase the processivity based on it. But work is ongoing, so work is there.
Oh, good, great, thank you. I think that was it from the chat. Does anyone in the room have a question for Ida? No, so then we will rig up Kalle here for his presentation. Okay, great, Kalle, that you could join us today. So Kalle is a researcher here at Uppsala University and you have a long experience from using Nanopore in many different projects. So we are very keen to hear about your experiences from Nanopore sequencing.
Thanks, Susanne, for the invite. All right. So today I will have a talk with the title Using ONT Sequencing to Gain Insight into Genome Structure and Function. And I will bring up 3 separate studies which all highlight different concepts of how ONT sequencing can be really valuable to extract information also other than the actual structure of the genome. We have the ballan wrasse, which is a fish species to the far left. We have rabbits, and we have Atlantic halibut. I will start with the halibut story. Sex chromosome evolution in a heterochiasmic setting. The Atlantic halibut is a really large flat fish species, which is also used in aquaculture in Norway and in other countries. And this is a research station a few hours outside of Bergen, where the Institute of Marine Research in Norway does some aquaculture studies on Atlantic halibut. And together with these guys at the Institute of Marine Research, we decided that there was a need to identify the sex chromosomes in Atlantic halibut because it was really relevant to know for certain aquaculture applications. But it's also highly interesting from an evolutionary genetics perspective because flatfish have highly dynamic genetic sex determination systems. We knew from before that male is the heterogametic sex. We knew that the genome size was about 600 MB and that we should have exactly 24 chromosomes. So we decided to do Oxford Nanopore sequencing of really long molecules of DNA and combine this with 10x chromium and high C, as well as transcriptome sequencing, both using Illumina short reads. And from a testis sample, we did ONT cDNA sequencing. The result of this the result of the sequencing using 10 million flow cells was that we achieved 31x coverage of the genome. And the reason we needed this many flow cells was that this was done some years ago. The throughput of these flow cells and the kits have increased since then. So the assembly the contig assembly software we used was Minimap2, followed by Mini ASM, followed by long read polishing using Racon. We used really high coverage short read data to polish using Pylon and ended up with a 611 MB assembly of 720 contigs. We bought the scaffolding service from the company Dovetail and were able to scaffold the contig assembly into 24 nicely looking chromosomes observed in the lower right in agreement with the karyotype information. One of the aims of this study, as I mentioned before, was to identify the sex chromosome. So we sequence short reads from male and female individuals as pools and compared the male to the female samples looking for divergent parts of the genome. As you can see in the delta allele frequency plot featuring all 24 chromosomes on top, we saw a clear increase in male versus female divergence on chromosome 13. And if we zoom in on chromosome 13, we see that actually most of this chromosome shows high divergence, which makes this compatible with it being the X and the Y chromosome pair. We did RNA sequencing, and when we compare male versus female gene expression ratio in the figure to the left, we see that only one gene is highly different in expression between males and females at an embryonic stage, and it's GSDF, which is also located on chromosome 13. And this makes this gene the candidate sex-determining gene. This was using RNA-seq, but we then started to evaluate GSDF expression across embryonal stages from 1 to 24 hours post-fertilization up to 13 days post-fertilization. And already from 48 hours post-fertilization, we notice a rapid increase in GSDF expression, specifically in males, but not in females. Using ONT sequencing, we could compare reads in the GSDF area, and we noticed that the X and the Y chromosome, they differ for gypsy transposable element insertion located just 2 kb upstream of this gene. And the haploid ONT assembly contains the TE on the Y haplotype, whereas the chromosome X lacks this insertion. And comparing with other flatfish species, we could also see that Atlantic halibut chromosome Y is the only chromosome carrying this transposable element, meaning it's derived in Atlantic halibut. We could also utilize another aspect of the ONT sequencing reads than just the structural information, because we called methylation probabilities across the region. And we could notice that the transposable element both contains a CpG island and is lowly methylated, which means that it could be able to act as a promoter for GSDF expression. And we did cDNA sequencing from testis samples of a male, and using these reads, but not the Illumina reads, we could see that there are chromosome Y-specific GSDF isoforms stretching all the way into the gypsy element. So these were revealed only by ONT cDNA sequencing. So our hypothesis is that chromosome 13 acquired the transposon, converting it through to a Y chromosome by altering GSDF expression. And this was because a gypsy transposon inserted close to the GSDF locus, which caused elevated expression of this protein, thereby converting these individuals to males. And we know that Atlantic halibut split from the Pacific halibut about 4 million years ago, which means that this represents a very novel Y chromosome. And we recently published this in PLOS Genetics. The next study I will tell you about is the BAL and RAS study, which is really a study evaluating the utility of PORE-C. Most of you are probably familiar with Hi-C, which is a protocol to retain cellular DNA 3D organization in a sequencing library by first cross-linking DNA and proteins, then using restriction enzyme digestion, followed by religation of the DNA. And this DNA will tend to be religated with other DNA, which is physically close, but not necessarily exactly the same fragment it was originally attached to. And this means that you save information about what is proximal to what in the nucleus. The traditional Hi-C protocol also has an enrichment step and an amplification step. PORE-C, however, is a tweaked Hi-C protocol to enable ONT long-read sequencing of one of these libraries. And there is also no need for PCR. In our efforts to evaluate the utility of PORE-C, we started out with tissue from 4 fish species where we needed Hi-C data, because we thought this could be an alternative to generating Hi-C data, because we get in addition to the contact information, we also get other types of relevant information from nanopore reads, such as methylation information. So we selected these 4 species, 2 of which I call species A and B, and 2 of which I disclosed the names, which was the European place and the Ballenras. And I proceeded with the PORC library generation using the official ONT PORC protocol with NLA3 chosen as the restriction enzyme. And it's a 4 base pair motif cutter. And as you can see in the tape station picture, as well as in the agarose gel to the left, the finished libraries varied greatly in size, with species A and Ballenras being much larger than the other 2 species. And consulting the ONT PORC protocol, we can see, which is shown here, what the finished library should look like. The species A and Ballenras resemble the expected output in a much better way than the other 2 species, which were probably over-digested. I decided to proceed with the Ballenras for a pilot experiment. And actually it was 3 sequencing runs done in this pilot experiment. Last fall, 2 runs of minion flow cells. And these libraries were generated without very stringent size selection and resulted in about 4 million reads with an N50 of about 1.5 KB with an estimated throughput of 21 gigs. Very recently, we started a collaboration with UGC to actually sequence one of these libraries on a Promethean flow cell. And I got this data just about a week ago. So I haven't had time to analyze this in the detail I wanted to for this meeting, but I did some analysis already. A difference is that the Promethean run was done, library sequenced on Promethean was produced using the LSK 110. And it was, the library had been subjected to a much more stringent size selection before sequencing. This run produced 12.2 million reads with an N50 of 5.8 KB, and you can see the size distribution of the obtained sequencing reads at the bottom. Altogether, we got 53 gigs of estimated throughput, and 40 gigs of these sequences were present in reads that were passed, in the past reads that created good base calling. And it really interesting aspect of PORC in relation to Hi-C is that a PORC read, which is several KB long, can contain many, many different submappings to the genome. And this is why I have this slide on combinatorics of contacts. So if you want to calculate how many pairs can be obtained from a PORC read mapping to 10 different loci, you use this type of calculation to come up with, you can create 45 unique pairs. So one read is actually equivalent to 45 Hi-C reads if it has this many mappings. And in the Promethean experiment, we altogether obtained 131 million unique contacts from these about 12 million reads. And the highest number of contacts in a single read was 87, which means it can be converted to about 3,700 pairwise contacts. And if we plot this, the number of unique pairs versus number of distinct mappings of a PORC read, we see how this increases with the number of unique mappings. And in this particular library that was sequenced on the Promethean flow cell, we see that we have 197,000 reads having 10 mappings, exactly 10 mappings to the genome. And if we compare number of reads in the middle plot to the number of contacts derived, we see that we get 8.8 million contacts just out of the ones mapping to 10 locations. And if we instead show it in this way, where we show read densities by read counts and read densities by numbers of unique pairwise contacts, we can see that we can notice the increased value of having long reads with many contacts in relation to just counting the number of reads. So if one wants to fully extract the added benefit of poor C over high C, one should probably do quite dramatic size selection before to retain mostly these reads that map to many, many locations in the genome. A very talented bioinformatics student of mine, Ines Borges, also started evaluating the potential to extract methylation information from these experiments. And she developed a pipeline to extract and visualize methylation profiles from the chimeric poor C reads. Methylation calls were made both with Nanopolish and with ONT software Guppy. And we also could show that low methylation methylated C densities increased with the proximity to transcription start sites, which is a hallmark of TSS profiles in the genome. And what you see at the bottom here is a subset of reads and whether they are likely to be unmethylated or methylated. And we can use this to potentially stratify chromosomal 3D contacts by methylation statuses of the interacting loci, which is a very interesting novel aspect of poor C in relation to high C. And now I think we have adequate data with the Promethean run to actually see if this is possible. In addition to using the poor C data for these novel purposes, we can of course also use it to scaffold genomes. And we previously generated a PacBio hi-fi assembly of the Ballan-RAS using hi-fi ASM. And when we extracted the pairwise contacts, as I mentioned before, we could feed them to a high C scaffolder, in this case, pin high C, to create a scaffolded genome assembly, which was then manually curated in JuiceBox. And this resulted in 89.8% of the assembly being scaffolded into placed chromosomes. And only about 10% of the assembly remained in unassigned contigs. So this means that poor C data can be used to scaffold genomes as well. And the last study I want to tell you about is Cas9 capture reveals a lowly methylated ERV in domestic rabbits as the candidate mutation for a fine map trait locus. So a previous attempt at a long-range PCR was done to define the structure of a candidate causal SV for a certain trait that I can't disclose today in rabbits. And we also attempted to do samplix capture followed by Phi29 amplification and ONT-based sequencing to define the structure of this SV. But in the end, we decided to just go with Cas9 capture followed by ONT sequencing. And to do this, we isolated DNA from blood of a heterozygous individual, which was wild type mutant, or it was heterozygous for these two alleles. And we used the Cas9 targeted sequencing protocol from ONT and 4 guide RNAs, as is recommended in the protocol. And we got a fairly high number of reads, which perfectly agreed also with the long-range PCR, which we later on actually managed to do, which means that we got the structure of the insertion and could determine that it's an endogenous retrovirus. We called methylation for each CpG site and could show that the trait allele and the wild type allele actually differed a lot for methylation exactly within the insertion region, residing quite close to a gene of interest that we think is the gene underlying the trait we investigate. And this is what it looks like when we, instead of showing the average methylation, show the methylation of each individual read in the genome, in the region. So in conclusion, ONT reads give value beyond just providing genome and transcriptome structural information. In Atlantic halibut, we could find methylation differences, and we could get the RNA-seq information about where the 5' of the sex-determining gene actually started. In BalanRAS, we analyzed the utility of PORC to show that we have potential using these libraries, not only to call methylation and to scaffold, but maybe also to contrast chromosomal contacts by degree of methylation of the individual contacts. And for rabbits, when we intended just to resolve the structure of the locus for free, we also got a really relevant methylation signature that we think actually could be highly relevant for the trait in question. That's it for me. Thanks.
Thank you very much, Kalle. Nice presentation. We have a question for you in the chat.
Yes.
We will take that one. So are you able to integrate methylation data in the PORC to further aid scaffolding or haplotype separation?
Maybe methylation could be used for this purpose, but I have higher hopes for actually the segregating genetic variants to actually leverage phasing in a completely different way than you can with Hi-C using the genetic variant.
Mm-hmm. Yeah, thank you very much. So yes, we will move ahead for the next speaker. Thank you, Kalle. So our next speaker is Melanie Tanguy. She is zooming in from the UK. She is associated with Genomics England, or she works at Genomics England, and I think Melanie is around.
I'm here, yeah. Sorry, just trying to find my windows.
Yeah, so great that you could join us, and feel free to share your screen. Good.
Okay.
Oh, great. Yeah, we can see your presentation.
Excellent. Good morning, everyone. So my name is indeed Melanie Tanguy. So it's very nice to hear somebody pronouncing my name the right way. Like I usually lose that in the UK for sure. So today I'm going to talk to you a little bit about like our effort to develop an end-to-end solution to basically do long-read whole genome sequencing in cancer. So I work for Genomics England, so I thought I would start there. So I don't know if you ever heard of Genomics England, but you may have heard about the 100,000 Genomes project. So basically at Genomics England, we are a public company. We are wholly owned by the Department of Health here in the UK. So the company was founded in 2012-13 to deliver that 100,000 Genomes project. So the sequencing of that project like reached its goal by the end of 2018. And what was interesting at the same time is that the NHS, which is our national healthcare system, NHS, at the time had his plan to build an NHS where access to secure linked clinical genomic and other data will support new medical breakthrough and consistent quality of care. So I'm keeping that sentence, which comes from our corporate slides, where I want to insist on this notion of delivering new medical breakthrough, but also a consistent quality of care. And I'll go back to that a little bit later. So what did that mean for Genomics England? So that's the beyond 100,000 genome project step for us. And that really means that we moved from a project-based company to a platform-based company, because we are delivering for the NHS this sequencing and data analysis. So what is really our goal at Genomics England? So our goal is to enable genomic healthcare and research. And one of our main partners is working with the NHS to embed genomics into routine healthcare. So routine healthcare for us, what does that mean? Well, actually in England right now, you can get whole genome sequencing as a routine diagnostic. So there are a few specific indications for that. So at the moment, the ones that are commissioned are for cancer, hematological tumor, pediatric tumors, and sarcoma. And there is a wave 2 coming soon that will include non-small cell lung cancer, ovarian cancer, triple negative breast cancer, and glioma. So there is also some indication in the rare disease space, and currently there are 20 indications for which clinicians in the hospital can select a test in the test directory of the NHS, and that test would be doing whole genome sequencing. So we provide the sequencing and the genome analysis for that. So we are also having 2 other goals. One that is enabling scientific research by our large genomic database. So again, what that means is that we're providing data, we are providing bioinformatic tools and the infrastructure to use this data and those tools in what we call the trusted research environment. So the third goal here on that slide is helping to improve diagnosis and treatments for patients, which is eventually our goal. We are a healthcare company. So I just want to stress here that that does involve what we discussed like so far, but that also involves enabling dialogue. So at the moment we have quite a big project about like newborn sequencing. So all these dialogues about consent, diagnosis, prognosis, and treatment are very important to us to help. So now I'm going to move to the long-read sequencing part. So this is part of an initiative called Cancer 2.0 that also involve what we call multimodal data. So that's how you combine genomics, pathology data, and radiology data. I'm not going to talk about that today. So this click. Okay, sorry for that. So this project is a collaborative partnership, and what we want to develop, demonstrate, and deliver is, well, what is the clinical utility of doing a long-read genome? Can it improve turnaround times? And can we build a distributed sequencing framework of clinically accredited pipelines? So we are working in the clinical space, so we want to reach that level. So our idea is to have like a sequencer in the hospitals, but then we still think that the analysis, interpretation, and data storage will be centralized. And alongside that, we are aiming to create research data sets that would be available for researcher, clinician, and pharma company to improve new discovery. So we have like a few stages in that process, from the sample pathway, sequencing workflow, down to accreditation and quality assurance. And today I'm going to talk to you about the 2 first steps. So we heard a lot about the advantage of long read, so I'm not going to insist on that too much right now, but 3 factors that for us are very attractive for using long reads is it can be rapid, it can be real time, it has to be easy, and it has to be cheap. We haven't talked about that too much this morning, but this is one of the key reasons as well. So if you want to implement that type of sequencing in the clinic, where do you start? Well, it's always good to start at the R&D stage. So I am part of the scientific R&D team at Genomics England. And what we do is technology evaluation, process transfer for clinical development, meaning that the transfer is done to the NHS in our case. And on the side, once again, our goal is to also generate these long native whole genome sequencing data resource for research. So where did we actually start? So we started very similar to NGI actually in 2018. We actually established a partnership with the sequencing R&D team at the Sanger Institute where we are hosted. So we are in Hingston, which is south of Cambridge, Cambridge being a bit north of London. And in the first half of 2019, we had our first PromethION setup. Yes, it took 12 months at the time to set up a PromethION, but it's much faster these days. And then from July onwards, we started to work on some pilot long-read whole genome project. We did a little bit with PacBio, but it's mostly ONT. And we use volunteers, so those were colleagues in Genomics England who accepted to give blood, but also rare disease participants from the 100,000 Genome Project. And we also use DNA Standard. So then, well, we had a little pause in April 2020, which was the start of many lockdowns in the UK. And the reason why we came back to the lab was to actually do a pilot on our COVID cohorts that we were sequencing in our accredited short-read pipelines at the time to try to decipher genetic bases for ICU, so intensive care unit hospitalization in COVID patients. So after that, we started to really work on this cancer project. We started last year, so it's pretty fresh still. So we had a phase 1 where we sequenced 100 cancer patients, I would say. So it's 2 samples. We do tumor and germline. And we usually run 1 flow cell for germline and 2 flow cell on the PromethION for tumor. And on the side, you can see a little representation of the number of people who worked on those projects. So we started, we were 3, 2 at the bench, 1 bioinformatician. We then had only 1 person at the bench and 2 bioinformaticians, and we started to grow from the cancer project. And since we started this phase 2, I have to say now it's a lot of people in the company. We really invested people's skills here, and we think it's going to be very helpful. So what have we done really? So this is a number of flow cells we've sequenced since July 2019. So you see it started quite slow. The flat lines are corresponding to lockdowns. And if you go, like that, so that was lockdowns, and then there are a few flat lines. That was the time we had only 1 person. Well, I was alone in the lab, and those are my early days. And you can see recently that we had really an uptick in weekly, monthly sequencing. So what do we sequence? So we had some development cohorts. We worked also on rare disease patients, a bit of COVID, well, quite a lot of COVID actually, and then all the rest is cancer. So we are sequencing those cancer that are already on the NHS list of possible tests with whole genome sequencing. So we did sarcoma, we did some breast cancer and hematological malignancy mostly. Here, I just want to attract your attention on these big blue blobs here. So recently, we also started to work on diversity projects. So we are working for healthcare. We are working in clinical setups. And it's absolutely key at that stage that we are increasing our diversity of genomic data if we really want to support equal access to the benefits of clinical genomics. So we're running this project alongside the cancer project. What are we really sequencing? So the samples we have in our hands are actually mostly biobank samples. the main biobank we are using is the one created for the 100,000 genome project, but we are also working with collaborators who may have clinicians who are created their own biobank for their area of interest. We dealt a little bit with fresh extraction as well. It's better. That was mostly for blood. And we also have a little bit of experience of long-read specific extraction, mostly for blood, but we again are starting to work with collaborators to see how that would work for tissue. So I'm going to now move to what makes a sample a good samples. And in the clinical space, I have to say good might not be what you've heard before. Suitable would be maybe the better word to use. So you've seen similar plots before. So this is the kind of traces you get from a machine called femtopulse. And this is going to tell you how big is your DNA. So what we like in our lab is to just bin everything that is under 10 kb, and everything above 10 kb we're happy with, but for above 25 kb we start to consider, do we want to shear or do we want to go for longer reads? These limits are moving. This slide is probably 1 year old, so I would like to switch that a little bit higher, but we are not there yet. So how did we come up with these numbers? So a little comment about coverage first. So that was some of our early development work where we were wondering, but how much coverage do we actually need? This comes from our germline analysis pipeline. So that was a rare disease minded exercise, and we don't have yet the data for tumor. But what you see here on the left panel is that if you are trying to recall a set of common structural variants on 2 individuals here, you see that when you reach about 30x coverage, you start to be in the saturation phase. So that's still our goal, reaching 30x for germline. And the second question is what kind of DNA do you want to sequence? And what we noticed at this early stage is that, so this is the same plot if you want genome coverage, x-axis, and here you have similar to that, the recall of common structural variant set. So you have one person here at the top, participant 32 and certified at the bottom. And what has been done here is that the reads of our sequencing experiment have been binned, so up to 10 kb in light blue, 10 to 20 kb, and so on and so forth. And you see that as soon as you reach the 10 kb plus 4 structural variants, we thought we would be in a comfortable place being at 20x and above 10 kb, hence our wish of getting rid of everything below 10 kb. So does that work with your sample? So that's again this pink, red line is the 10 kb limit. And you see that when we receive samples, this is a good one, by the way, for us. So you have a high molecular peak on the right here, but we often see a little bit of degradation. So as I say, we tend to bring that high molecular peak to something a little bit more manageable for the sequencer. And very importantly, we're trying to get rid of these short fragments. I just want to show you here the impact that does have. So we sequenced these 2 samples here. We got a similar yield in both cases, but the N50 was drastically different. So by doing this extra step, which is extremely costly in terms of sample, you still get a lot better out of your sequencing run. So how does that work in real life? Well, actually, that's samples we have to deal with. This is still these 10 kb lines. And you see that if you would like to remove everything below 10 kb in those cases, well, you have no more samples. So sometimes we do have to sequence these samples. We are trying to generate evidence for clinical use. So we are sequencing what we have. So it's not always beautiful, but that's what you can do. So we also have very good clinical samples coming from collaborators. So it is one we managed to reach like N50 or 25 kb, and this is still from biobank samples. Okay, so next, I wanted to talk to you about what we do with the sequencing data when we have it. So what I'm going to present you here, and I've already like done, is maybe the difference of what we observe in real life compared to what you can see in Nanopore presentations, and also in some kind of flashy announcement on Twitter sometimes. So that is a real life experience of the mean aligned read identity that we see. This stops last year because I don't have the updated plot, but you see that there were like massive improvements in Guppy, so that's the base caller of Nanopore, and mostly like between 3 and 4, it was really impressive. And I think this really keeps improving, so it's very important to keep on track with the latest advance. There is space to improvement here, and I'm confident that we will reach like these Q20 that are promised, but we are not there yet. Once we have this base called data, or germline pipeline works like that, we are using Minimap2 for the alignment, and then we run like a few algorithms, mostly SNF to call structural variants. We are currently also focusing on our QC pipeline development, for which we are using HLALA, and getting some base calling stats coverage information. So in terms of infrastructure improvements, I think what was really key for us is to go from this V1 where everything was done locally, base calling on the machine, to where we are now, where we only use the P48 to do sequencing, and all of base calling, QC, and analysis is done at Genomics England. And this is really going to this V3 infrastructure that what allowed us to really have this uptick of sequencing that I've showed you on the first graph. So upcoming for us, it's working a lot more on the sample pathway, especially for tissue. We are going to keep sequencing to create this nice sequencing cohort that will allow us to do tool benchmarking at the moment for SV and methylation only. And we are starting to generate evidence for clinical use cases. In our pipeline for future development, but coming soon, we want to work a bit on automation and standardization, clinical validation, and trying to introduce all these nice technology upgrades that keeps coming with Nanopore. Work a bit on the integration with Illumina, and keep pushing for more than SV and methylation in terms of copy number alteration, indel calling, phasing, and so on and so forth. So there are challenges, but there are constant improvements, and we are still very confident that in the next few years we'll get there, or having clinical Nanopore sequencing in the clinic. But the sample source and quality is really key. And I would say you have to be flexible to stomach all the advances that make things better. One last thing I wanted to say is like it takes like a full ecosystem. You need like all these colleagues who are not only bioinformatician, but software engineer, platform engineer. You need the analytical tool set to grow. We are relying heavily on effort from academics to provide those tools. So I think we need to recognize that. And what we really would like to see is a bit of stability. So you have to balance like what is the new hot things coming and how you can generally use it in the clinic. So we are hoping to have a lot more to say to you in a year or 6 months, hopefully. And I would like to finish by thanking the team, especially all these people who are working in the lab. Adam has been basically creating all this pipeline from scratch. So a big thank you to him. And the lead of the new teams that started to work on Concert 2.0, and also the Sanger for hosting us. But maybe above everything else, we have to thank like our participants from the 100,000 Genome Project and those patients who are signing up for research and accept to share their sample with us. Thank you.
Thank you very much, Melanie. Nice to have you around soon. Sorry, just speak closer to the microphone. Now you can hear me. So do we have a question from Melanie? Yeah, we have one question in the room. So speak up clearly. We have the microphones in the roof, but
okay. Let's see if it works.
Yeah.
Do you hear me? So thanks for a great presentation, Melanie. What I would be interested in is like how many sequences are you running? Or maybe I should. Okay, let's try it like this.
Oh, yeah, that's right.
Yes, so thanks for a great presentation, Melanie. It's really impressive, so the amount of sequencing that you're doing. But could you say something about sort of how many flow cells can you run a week? And
yeah.
Also, is there like a target for the cost that you're trying to approach here?
Keep. So yeah, so really when we stop, so when we were doing base calling on the machine, so we have a P48, so that's the P48 with V100 inside. So you are kind of limited to 16 at one time. We run for 3 days, though the third day in terms of sequencing is not very useful for anything, but it was useful to keep up with base calling. So now we do the base calling offline. Well, you know, the limit is the number of slots on your P48 that are actually working. So I would say 48, maybe not always. And in fact, at the moment, we do 64. So we have 2 P48 plus 1, but we are supposed to return it soon. And we do, yeah, 2 batches of 32. This is really only the 32 is because we work in strips, and that works as a multiple of 8. We seem to be keeping up okay with that at the moment. Sorry, just kicked me out. And our objective is to reach about, we do that twice a week, so we are at 128 per week at the moment. The pipeline is a bit screaming, so the people running the pipeline right now are not very happy with what we can do in the lab, so that's why we really need automation. It's impossible to push by hands like all your sequencing run when you do that many.
Yeah, and it has to be an incredible amount of data also to.
So Adam was telling me yesterday that we have, I think I had it on one of my slides. I'm not going to, sorry, I get it so big. Yeah, we have 1.5 petabytes of data so far. So the release on our research environment is also going to be interesting. It's a lot of data to deal with. We are keeping all the fast file because of all these improvements in base coding. At the moment, we think it would be crazy to not store the fast file. So we are hoping that if needs be, we could like rebase call and get better value out of it. Obviously, methylation.
Yeah, and I guess the final thing is perhaps the cost then. I mean, you say cheap, but is there's no number for what a patient should, how much it should cost using this assay?
So in the clinical space, like the cost effectiveness is going to be decided by the NHS, and they have an organization that deals with that. What I just want to say is like yesterday we were checking quickly on the test directory for I think ALL. If you look at the number of individual tests that this patient could have run for their conditions, and you think that one whole genome sequencing could just cover all of these tests in one go, then the cost efficiency can be, it doesn't need to be mega cheap, it needs to be competitive with what exists. And what exists currently is Illumina. I guess we need to compare it with that. So, I mean, there are like some commercial sensitive information there. So, but as long as Nanopore like stay on par with Illumina or get cheaper, I think they're going to be in a good place. They can't get more expensive.
Okay, thanks.
Yeah, thank you. Yeah, we have another question here. Yeah.
Yeah, thanks for a great presentation. So you showed that sometimes you do the QC of the samples that you get in, and sometimes it looks great and sometimes it does not. And I'm wondering how much influence can you have on how the samples are extracted and treated before they get to you? Do you have an influence on that process at all?
I wish. So yeah, so we are so influence. I think it's a collaborative partnership, so it's like all together. The thing is that you have to think about like a clinician in an operating theater. We're already kind of trying to keep the momentum for them to not throw everything in formaldehyde and do FFPE samples everywhere. So there is another question about like, do you actually want to ask them to use kits for specific for long reads? What we really want to focus at the moment is like to really assess their current extraction methods to see if actually they could be okay. We have good experience with basic, without citing any brand, but CADG and kits, where actually if you handle the sample quite well and you don't do like 50 free flow cycles before sequencing, actually it's good enough. And you know, if anything, then we don't have to shear. And if we don't have to shear, we win 20 quid per sample. These shearing columns are like insane. So, you know, I don't think there is a space for ultra long read for research, especially if you want to do genome assembly or de novo genome assembly, but maybe for really clinical use, this is not what we are going to be shooting for. And yeah, wait and see. We are really starting to work with real life samples, so we'll see. I'm hopeful.
Thank you.
Yeah, thank you very much. Do we have another question? No, and I cannot see any questions in the chat either. So thank you very much. Oh yeah, we have one here. Yeah.
Hey Melanie, this is Jakob from ONC. Thank you very much for a nice presentation. I was just wondering, with regards to the diversity sample cohort you had, it seemed to be quite a varying coverage you achieved from those. So could you speak a little more to the sort of sample differences or qualities, or what the reasons behind those are? I assume those are biobank samples as well.
Yes, biobanks are already extracted, so it's not biobank tissue or blood. So actually we are going to change our cohort and we are hoping to increase the sample quality. My first reaction to your question would be, you tell me why I have such a bad coverage. But obviously your colleagues here in the UK are on it. Yeah, they saturate a lot these samples. I don't know. So when I say saturating, meaning the pores get saturated and then you cannot recover it, even if you do nucleus flush, which we do, by the way, all the time. I don't know. I think, well, actually, I've heard a presentation from somebody from Uppsala, I think a few months ago, about how samples like quality on Nanopore for like various sources like affects the sequencing quality and output. And it's just crazy. Nanopore is native sequencing, right? As you know, and what you put in is what you sequence, and sometimes it doesn't work. When I talk about stability of the process, this is what we are after. How can we guarantee that we have 30x coverage reliably on one PromethION flow cells? That would be really good, at least to predict when we want. You know, ODs look fine, yet you put your sample in and then you get 50 gig. We had like full cohorts where everything like was like above 30x coverage. So for me, that's also where we are going towards. And it's getting so much better months after months that I'm sure we'll get there. We're just not there yet. And I don't know why this particular core didn't sequence very well. But if that can be of any joy for you, it did not sequence very well on other platforms either.
Thank you very much.
Yeah, thank you very much. Yeah, I think we already moved into the Q&A session here, maybe. So we do have a few questions in the chat that I think I would like to ask you. So the first one is actually for Jakob. Are there any attempts to concatenate shorter RNA species to optimize output, read depth of ONT long sequencing reads?
Could you repeat, please?
Yeah. Are there any attempts to concatenate shorter RNA species to optimize output or read depth of ONT long sequencing reads?
Not that I know of, and I'm not entirely certain. So so Claudia, what do you mean about optimizing or maximizing the output?
So you mentioned, can you hear me in
Yes.
Uppsala? So you mentioned that you have now developed that you can now basically have bases from 20 to 500 million. So you could basically also take all the shorter RNA species that are processed RNA species in the transcriptome and concatenate them. So those ones that are like, microRNAs that are about 20 nucleotides or tRNAs that are about 70 nucleotides in length, if you were to concatenate them, then you can have a long sequencing read and thereby maximize to assess, for instance, the abundance of these RNA species or the modifications.
Sure, got it. Thanks. Okay, so if this is on the direct RNA platform, it's going to be slightly different because the direct RNA kit currently only works on poly-A tail material. So so one would need to concatenate the RNA species and then add a poly-A tail or something in order for that to work, as far as I can see right now.
Okay. Yeah.
But yeah, so I don't know of anyone who have done such a thing, but it doesn't mean that no one has necessarily. So, but yeah, I'm afraid I don't have any really good answer to that right now.
Thank you.
Yeah, thank you. We will move on, Jakob. We have another one for you here as well. Is there any upcoming kit or recommended protocol for library preparation for metagenomic sequencing with the aim to sequence RNA and or DNA viruses in human samples? Also, is adaptive sampling well suited to remove human sequence?
That's another really good question. So I did see an adaptive sampling approach quite recently to remove host DNA reads and just concentrate the sequencing on the metagenomic part. So I think there's nothing special about the actual library prep. So you would just use the standard kits for that, as far as I know. And there was something else to that question, wasn't there?
Yeah, adaptive sampling, well suited to remove human sequence.
Yeah, so there are at least some early efforts into that, and a lot of users are playing around with the capabilities of the adaptive sampling. So certainly it's something that I expect to mature even further with you know, all the great user work going on, and also further developments from Nanopore, of course.
Yep. I think. Thank you.
Maybe on that note, we are, as communicated at the Nanopore community meeting as well, we are looking to enable adaptive sampling on the PromethION system as well. So in order to achieve higher you know, coverage, etc., that's probably going to be an opportunity at some point through this year as well.
Yeah, good. So we have a question for Anna here in the chat. So Anna, are you looking to increase GPU capability to standalone machines, with standalone machines, or would a grid ion be a possible alternative?
I think it's also a question for Susanne, perhaps, but I think we don't have any plans to buy a grid ion.
No, not right now.
No, I mean ion sequencing is something that individual labs can do, and for us, PromethION is more like relevant. But I would like to add actually to a previous question,
yeah, sure,
about RNA, about polyadenylation of total RNA. One of our groups that we have worked with to sequence direct RNA tried that once in one sample, and the sequencing was really bad. Actually, it was kind of horrible, to be honest. And it's also hard in that sense to assess the input RNA, because when you assess input RNA normally, thanks, you would go for like ribosomal peaks, and this you will not see if you have polyadenylation before you assess the input RNA. But this is like, it's theoretically possible to do. It just we kind of tried it once and then never again. But who knows? Maybe we will at some other point. But back to GPU, it's mostly now for PromethION.
Yes.
Yeah.
Yes. So we move on with the question to Ida. So do you happen to still have some of the 80 terminated reads available?
So me and Olle, we already spoke about that in the chat.
Okay.
He's interested in looking at them, but I just want to clarify that actually all the amplicon sequencing we did in that project was with PacBio and not Nanopore sequencing. And he was interested in the Nanopore reads to look at some biases. But I still have the reads though, but I don't think he's as interested in them anymore.
Okay. Understood. Yeah, I think that we have discussed all the questions that are available in the chat, but we have one more question in the room.
So I have a question for Anna, who I'm sitting right behind. Yeah, so you mentioned that you use around 5 micrograms as input for the ligation prep, right? Which is 5 times higher than what is kind of ballparked by the official ONT protocol. So I'm wondering whether you're concerned with any reagent depletion.
So 5 micrograms is not what we do for library prep, it's what we ask from the user. And that is to be able to first eliminate short reads and then do shearing. And depending on the fragment length, you lose more or less while you shear, and also purify with amper beads. And then also, it's also always good to have extra. So we say we want 5 micrograms, and that is if we want to rerun stuff, and like if the DNA doesn't work, we want to try again. But like I also said, if you don't have 5 micrograms, we can solve it.
Yeah. So another question in the room here? Yes.
Question for Jacob, actually. So with the newer flow cells that have a higher accuracy, I assume it comes sort of at a cost in terms of output or something. Otherwise, everyone will be running only that. So what are the differences between the R9 and R10 in terms of output?
Yeah, that's a really good question as well. So maybe first of all, it's not down to the actual nanopore type. So the R10.4 nanopore will perform equally to the R9.4. So it's more related to the library chemistry. So right now we do see around 60% of the yields obtained if you apply the kit 12, aka Q20, compared to the exact same sample using kit 10, for example, LSK 110. So there is that drawback right now, but Clive did tweet something not too long ago about different. So the reason for that is because the motor enzyme runs at a slower speed as compared to the production kits right now. But Clive did allude to that they've tested a number of different motor enzymes for the Q20. So I assume that's what he's going to speak about this afternoon as well. So there seems to be some upgrades along the way. And I mean, as with everything Nanopore, we continuously improve on these things. So chances are you're going to be getting Q20 or Q30 in not too far future at the exact same yield that you would be getting now on kit 10 and R9.4.
Yeah, thank you very much. More questions from the room or on the... No? Then it's already past noon, so it's lunchtime. So I think we will close today, and I would like to say thank you very much for attending today's seminar. And thank you to the speakers who joined us, both here and via Zoom. And yeah, have a great afternoon. Thank you.